Okay, so topic 9 today, we're going to talk about enzymes, and hopefully we have time at the end. I have a Kahoot for you that will cover a little bit of topic 8 and a little bit of topic 9. So just a reminder there to get your app ready near the end. And yeah, so let's talk about enzymes. So first thing, it's just a reminder about last day. We were talking about enzymes, and I had mentioned that we're kind of mostly just getting ready for talking about respiration and photosynthesis, which involve a whole bunch of enzymes. And these pathways have quite a number of reactions. Glycolysis, for example, is actually 10 chemical reactions, and each of those reactions are catalyzed by enzymes. So it's worth it to talk a little bit about enzymes. What are they and what do they do? So you probably know that an enzyme is a biological catalyst. So what does that mean? A catalyst is something that speeds up a reaction, and it does so without being changed itself. So here's an idea of how an enzyme might work. So we talked about penicillin before, once or twice, and we're going to talk about it again today a couple of times. And I'll show you what kind of speeds we're talking about. So penicillin, you may know, is a drug, and we use it to kill bacteria. And if you're sick and you have an infection, you might get some penicillin. And the drug will last in your body for quite some time. It does break down, though. You can see that this is penicillin G, and the half-life of penicillin G is it's about 84 hours. So that's like, what is that, 4 days, something like that, 3.5. It doesn't really matter, because if you think about it, bacteria, oops, sorry, PowerPoint's doing funny things on me here today. The bacterium, their lifespan is maybe 30 minutes. So if the penicillin is in your system for a few days, it's enough to hopefully clear the infection and kill all the bacteria. Now you probably know if you get an infection, you're usually taking the drugs for a week or 10 days, and that's just to make sure everything clears. But in terms of the lifespan of the bacteria, the drug is around long enough. Now, you probably also know that some bacteria have resistance, and some of them have an enzyme. And that enzyme, there it is called penicillinase. And this is a very fast catalyst. So how fast, you're probably wondering? That's how fast. Penicillinase can degrade, sorry, I don't know what's going on with PowerPoint here. Something wrong with my mouse here today. Here we go. Penicillinase can actually degrade up to 1 million molecules per second. This is super fast. And so this is actually how some organisms get resistance. So this is trying to show you how fast enzymes are. A lot of reactions are too slow that would happen biologically. I'll show you another reaction. This is a reaction of something that you might eat. So sucrose is table sugar. And if you have sucrose in water, the half-life of that glycosidic bond is something like 100 years. So that's right, 100 years. It'll sit around and basically not break. Now, we don't have 100 years to digest our food. Some of us certainly don't have that long. So we have an enzyme called sucrase that breaks it down. And the sucrase breaks it down to glucose and fructose and does it in milliseconds. So that's very fast, and that's kind of very useful because, of course, we want to live and be able to do things. So a couple of things about this reaction. This is kind of convention. Usually we start over here, and you have all the substrates listed on one side, or substrate in this case. You have a reaction arrow, and the reaction arrow will have the conditions of the reaction. So in this case, it has the enzyme. If this was a chemistry lab, you might say the temperature or solvent or something like that. And then you have the products written on the right. Often we'll also give the delta G. So bringing us back to last day, what is the delta G again? So delta G is the free energy, and you're probably trying to remember, okay, this is a negative value. What does that mean? That means that this reaction is thermodynamically favorable, and that means that the reaction is going to occur and it doesn't need energy to proceed. Doesn't mean it's a fast reaction. Naturally, it's a very slow reaction, and this is why we need enzymes, because even though we may have reactions that are favorable, they just may be super slow. So what we want to do is speed things up so that we can have things useful in our body, particularly if we're trying to consume food. So a little bit more about enzymes. I'm going to talk a little bit about what this means about lowering the activation energy. But most enzymes are proteins, and you can see there's the enzyme there. It's those blue things are atoms in a protein, and it's taking a substrate in, and it's taking it into the active site where the reaction is going to occur. So we'll talk about the active site in a minute. Let's talk about activation energy. So our textbook is using this symbol here to represent activation energy. Activation energy sometimes is represented in textbooks in different ways. I've seen E in a big A, I've seen A in a big E. I think in my chemistry textbook it has something like delta G with a funny star in front of it. So we're not going to worry about the symbol, we're just going to call it activational energy. So what is activational energy? Activational energy is the amount of energy just to get something going, get something started. So think of it like you have a pile of wood and you're making a campfire. It's not burning until you give it a spark or a match or something like that. And that's what activation energy is like. It's a little shove or a little spark or a little match to get something going. Another way I kind of think of it as is, you know, it's winter, and let's say I'm making a giant snowball, and you know, I see somebody down the hill that I want to, you know, the snowball to roll after. I got to give it a little shove to get the whole thing going. It's going to roll down the hill. So if you take a look at this reaction pathway, what they're showing over here is we have the reactants at the beginning, and we have the products at the end, and the reactants have a higher potential energy than the products, so kind of like the diver on the diving board, and as the reaction occurs, it's going to lose energy, and so this delta G is negative, right? So it's less than 0, it has a negative value. So that's thermodynamically favorable, but to get going, it has an activational energy, which may or may not be huge. And sometimes it's hard to get things started. You know, you might take 2 matches or 5 matches, or, you know, in the case of some people I know, to get their campfire going, they're putting gasoline on it. So that can be a barrier to get things going. And that's why we need enzymes. So this is showing that same graph, but showing what enzymes do. And enzymes reduce that activational energy. So what I'm looking at is right here. It's a lower activational energy than the natural activational energy. So delta G is unchanged. The overall energy of the reaction in terms of how much is released or consumed in the reaction is not changed, but the activational energy is, which means it just needs a gentler shove to get going. So let's talk a little bit about that. What I do have for you, though, is I have this this little video. It came with the textbook. It kind of needs to be updated. It's kind of low quality, but it basically explains what I said, and I think he does it just a little bit better than I do. So I'm going to play that for you right now. It's only about a minute or 2 long.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its 3-dimensional shape allows it to act only on specific molecules, referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy.
Oh, I don't know what happened to my video there. Looks like it's not functioning, but anyway, hopefully you get the idea. I kind of want to talk a little bit about that activational energy stuff in a minute. But before we get there, I want to just talk a little bit about chemical reactions in general. So it turns out there's actually 6 types of chemical reactions, in case you didn't know that. There's actually a committee. There's always these committees, these international committees of, what's it called? International Committee of Enzyme Classification. Probably a really fun job. Haha, no, probably not. But anyway, I'm not going to ask you about the 6 classes of enzyme reactions, but you can see there's a description here of them. And you may see them in your travels, particularly if you take some biochemistry classes in your future. You're going to definitely talk about these different classes of enzymes. And you might see a few of these names this semester as we talk about a few different reaction pathways. So one thing that is important to know is that enzyme names usually end with A's. And usually in the name, there's some sort of description, often the substrate or the type of reaction. So here's some examples, right? So a hydrolase is an enzyme that is involved in hydrolysis. So you know what hydrolysis is? That's where we're breaking a bond and we're using water to do so. You can see the next couple, DNAase and proteinase. The name of the substrate is in the enzyme name. So DNAases, of course, break down DNA, and proteinases break down proteins. And there's lipases, which break down lipids, and so on. Some of these things are named after different types of reactions that, if you know your chemistry, it might make sense. But if you don't, like a lyase, that's a removal. That's a breaking a bond, but it's not a hydrolysis reaction, so it's a little bit different. Don't worry about that one. Oxidoreductases, so it's right in the name. It's some sort of oxidation reduction type of reaction. There are transferases, meaning that it's moving a functional group from one molecule to another. Isomerases, that means they're rearranging the atoms, so making one thing from one isomer to another. And ligases. So ligases, we're going to be talking about that one when we talk about DNA replication. And so that's a few units from now. I think that's topic maybe 15 or 14 or something like that. So no need to worry about ligases yet, but notice they all have A's on the end. And like I said, some sort of description. And if you're good at naming your enzyme, the description is going to be really good. So what do enzymes do? Like I said, 6 different types of reactions. We're going to see a lot of things either broken down. So you can see this is the case of sucrose getting broken down into glucose and fructose. And there it goes. I don't know why I have the numbers on there. Or it can do the opposite. It can build things up. So in this case, you can see, I'm not sure why this is all blocked off, but we've got 2 substrates and they're building one final substrate. There's a little animation for you. I don't know why it's not playing. Oh, there we go. It's playing now, and you can see the 2 substrates getting together in the active site, and they're departing as one product. Just waiting for the animation to finish up. There we go. They also transfer things, and this is what you see in a lot of biological reactions. You see these transfer reactions. And if you take a look here, we've got some sort of functional group B. B is attached to C. It goes through the enzymatic process, and now B is attached to A. The most common type of reaction where we see this is the transfer of something like a phosphate group. So if you take a look at this reaction here, we've got a phosphate group. It's attached to our substrate, and we've got ADP over here. And so we take that phosphate group, and it's being added, and now we have ATP. So that's a transfer reaction, right? Where you have a bond being broken, but a bond being formed, all kind of in the same step. So enzymes, like I said, are usually proteins. They're usually large molecules. Some of them are very complex. Here's an enzyme we're going to be talking about in the next unit. This is called ATP synthase. So you can maybe guess from the name of the enzyme, ATP synthase, it synthesizes ATP. And this one is super complex. It's got something like 15 to 20 subunits, and it does a whole bunch of things, and we'll talk about it very soon. So the active site, as I mentioned, that's where the catalysis takes place. You can think of the active site as like a mouth. It's a binding cleft is maybe a better name for it. And what the active site is doing is it has amino acids there, and those amino acids are going to have complementary charges to bind that substrate. So I'll show you an example. Here's an example. You can see we've got this substrate here. This substrate is a carbohydrate called RUBP. That's found in photosynthesis. And notice, for example, we have an amino acid here, histidine, and that histidine has a complementary charge to that oxygen. So the positive charge, the hydrogen has a partial positive charge, and the oxygen has a partial negative charge. Maybe it's a little more apparent here where we have definitely a positive charge and a negative charge. And so that binding cleft, the active site, is going to have a pocket that's the right size and the right chemical forces to attract the substrates for the catalysis to happen. I know I have some other pictures of some active sites. Here's lysozyme. Lysozyme, this is an enzyme that binds peptidoglycan. So this will actually break down the bacterial cell wall. And it turns out you have lysozyme in your eyeballs and your blood. And this is actually protecting you against certain bacterial function or bacterial infections. So I was showing lysozyme for one other interesting reason is notice it doesn't end off with A's. So you're probably wondering, I thought all enzymes ended with A's. No, I said most of them do. This is an exception. This is like the first enzyme ever discovered. And so there was no formula for naming enzymes back in the day. So it ends up having its own name. Here's another active site, and this is the active site in a DNA polymerase. And DNA polymerase binds DNA and makes two daughter strands. And you can see we've got like a pink daughter strand right here and a green daughter strand right there. And anyway, you can see it has kind of a unique binding cleft. It looks a little bit like a hand reaching out and grabbing DNA. So we'll come back to this one here, rubisco. We're going to talk about it with photosynthesis, but at the moment I just want you to take away that there are complementary intermolecular forces in the active site that are helping to bind the substrate or substrates. So this kind of leads us to that question about how do enzymes do what they do? How do they lower the activational energy barrier? And usually in class I have a little bit of a demonstration. I have some things that kind of lock and click together. It's not so easy to do virtually, but I do want to write a couple of notes for you about how do enzymes do what they do. And I think I'm just wondering, I'm trying to remember Chemistry 102, whether this is talked about a little bit in Chemistry 102. But there's basically three things I want to talk about. One is called the orientation effect. Orientation effect. There we go. So what does that mean? It basically means we bring substrates closer together. And hopefully that one is obvious. So brings substrates close together. So that one's probably the most obvious out of it all, right? If you have two substrates, they're not going to react if they're not in the same room together or not in the same not in the same ballpark. Sorry, I meant to call this the proximity effect. I'm getting ahead of myself. Proximity effect. Sorry about that. The second one is called the orientation effect. And it's kind of related to the proximity effect. Orientation effect. It's Monday, so please forgive me for getting mixed up on a Monday. Orientation effect, it means that the substrates are rearranged or oriented. Orients the substrates. And it basically rearranges them so that they can react. So picture you're trying to attach two things together, right? Let's say you're trying to plug in something into the wall, into the electrical outlet. If you're bringing that plug and you have it sideways, it's not going to plug in, right? You have to turn it and you have to make sure that the little knobs line up with the little holes in the wall, and that's what the orientation effect is. So like I said, these things are kind of related. It brings them close together and brings them together in the right arrangement. Sometimes textbooks will throw these two things together and sometimes they put them apart. I usually describe them as two different things because both are definitely important in the catalysis reaction. One other thing, and you can see it's sort of represented on this slide. I couldn't find a really good diagram of this kind of thing. The last thing is something that's a little harder to really talk about without spending almost an entire lecture on it and going through some advanced chemistry. But I'm going to call this providing the reaction with alternate pathways. So providing alternate, and I'll give you a couple examples of this in a moment once I'm done writing. Providing alternate reaction pathways. So if you take a look in B, it's not really shown very well in that diagram, but there's a couple of ways to do this. So you may remember last day we were talking about how some reactions require energy. So sometimes that's part of it, is we energize the substrate. So what does that mean? It means we change the substrate before the substrate is being reacted. So somebody's asking me to go back, I'll do that in a second. And the other is, and this is maybe a little bit easier to understand, and I have maybe an animation, I'm hoping it will work on the next slide, is bending bonds. So if you think about this, you know, sometimes when you're trying to break something, I'm just trying to think of a good example of something that you might be trying to break. Let's say you've got a little metal tab on a food container, and you know, you can either just try pulling it off, which might not be so easy, or you might just bend it back and forth. And so this bond bending, I thought that was shown in this diagram, maybe it's shown in one of the previous ones, is another common way that these pathways are done. And like I said, it's a little easier to demonstrate in person. Oh, there it is, yeah. So there's the bended bond. You can see what it's doing is flexing that bond, and it's kind of, you know, wiggling the whole thing apart, right? So you can sort of picture it that way. I'm not going to go into these in any more detail. Like I said, it kind of takes a bit of a whole lecture to really talk about it, and we're talking about a little bit more of advanced chemistry. But you know, kind of an introduction here in this class anyway. So there's the other note, by the way. Hopefully you got that. I'm going to move on in just a second. I'll give you about 5 more seconds, and then I'll move on. Okay, so yeah, so I'm going to try to play this animation. Sometimes it doesn't work for me. I'll play this in a second, but back to lysozyme. So lysozyme is trying to break this bond here. Oops, this bond right here. So it's a hydrolysis reaction, and you can see the water is added over here in a later step. So one thing about this bond, as I mentioned before, is there's all sorts of hydrogen bonds that are stabilizing peptidoglycan, kind of like cellulose. There's hydrogen bonds all over the place. So you can see what's going on in the enzyme is that the carbohydrate is kind of getting warped out of shape. And what that does is it kind of loosens up the hydrogen bonds and then allows the water to, you can see the water's right there, to kind of get in there and break the bond. And so I'll see if this animation plays for you. It's kind of low tech, but okay, it says the internet site is not found. So, okay, sorry about that. I was looking for a good example, and the animation was pretty decent, even if it was low tech. But you can see in this diagram some of that bond bending that's going on. All right, so moving on, we have some more important things to talk about enzymes and a few other things to kind of define around them. So one thing you may have heard of is these things called cofactors or coenzymes. And a lot of enzymes have parts to them that are not amino acids. So for example, you may have heard of hemoglobin. Hemoglobin has a molecule called heme attached to it, and that's actually where the oxygen binds. Some enzymes have metals, so all of these things here, calcium, magnesium, zinc, potassium, these are all important parts of our diet. And sometimes they are essential for certain enzymes to function, which is why we have to have these things. A lot of these things are vitamins. You can see like biotin. You can get biotin supplements, NAD, riboflavin, which is vitamin B2, and coenzyme A, and things like that. And like I said, these are often essential for the enzymes to function. So if you see that term, I'm not going to really come back to this term. I want you to know what the term cofactor means. Don't worry about the other terms in this class. But that's usually a multiple choice or true or false kind of question on one of the midterms. All right, so test yourself. True or false? Enzymes change the amount of free energy required for a reaction. So there's those thermodynamic terms for you. What was free energy again? So the answer is false. So remember that the delta G is not changed in a reaction. So enzymes, if we want to change this to be correct, we would say enzymes change the amount of activation energy required for a reaction. There we go. What about the second one there? True or false? A cofactor is made of amino acids. The answer there is also false. That was just on the previous slide, and it basically said that it is a non-peptide component. So we could change that to not made of amino acids. So moving on, I want to talk a little bit about inhibitors and enzyme regulation and a couple of other things like that. So enzymes are relatively picky biological entities. And that's one reason why, as organisms, we need to maintain kind of a relatively constant temperature, because if we get too hot, our enzymes are going to denature. If we get too cool, our enzymes aren't going to work properly. You can see this is showing the right here. This is the optimal temperature for a human enzyme. So body temperature is somewhere around there, 37 degrees. And that's where most of our enzymes are best functional. A certain temperature, by about 50 degrees, they start to denature. As things get cooler, they're not functioning very well. And that's pretty typical of all enzymes, is that they have optimal temperatures where they function. You can see alternately on the right, there's a heat-tolerant bacterium, and so it's adapted to living in hotter conditions. So that's why it's over there with higher optimal temperatures. On the bottom, you can see it's talking about other conditions like pH. You can see we've got stomach enzymes here, and intestinal enzymes there, and those are environments that have different pHs. So the enzymes that are found there have different optimal temperatures. Enzymes sometimes have to be localized in specific organelles in order to function properly. So this is an example of mitochondria, where mitochondrial enzymes aren't active until they're actually in the mitochondria. So as I mentioned, I want to talk about regulation for a few minutes, and I have something I'm going to draw for you in a few minutes as well. First, it's important to talk about something called inhibition. So inhibition, so to inhibit something means that it is stopping something. So when we talk about inhibitors of enzymes, there's kind of two types that we classify them as. So if you take a look here on the left, we have an enzyme with normal binding, and its substrate is binding the active site. So some inhibitors are competitive inhibitors. So if you take a look at this inhibitor here, it's binding the active site and blocking the substrate. So when that happens, the reaction is not going to occur because the active site is entirely blocked. Some inhibitors fall into this third category, which are non-competitive. So you can see that this inhibitor is binding a different part of the enzyme, and it's affecting the active site somehow. So it's kind of like, sometimes I think about my hand, right, as being an enzyme, and my hand is, let's say my hand is grabbing onto, let's say a baseball or something like that. And then someone comes behind me and they, let's say they pinch my wrist and my hand loosens. So it's kind of the same idea. It's something on the other part of the enzyme that's affecting the shape of the active site. So I have a little video to show you that explains this, and I'll play that for you. Once this loads up, here we go. And it talks about the different types of inhibitors.
During the normal enzyme catalytic cycle, the substrate encounters
Hold on, I need
a specific active.
Replay that with the volume this time.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as a non-competitive blocker is bound to the enzyme.
So that's inhibition, and I want to give you guys some examples of some inhibitors and talk about something that's going on in glycolysis. So a lot of inhibitors, by the way, are drugs, and they're useful pharmaceutical agents. So here's an example of a drug called AZT. So AZT, you can see it says here, it's a nucleoside analog, and this is actually the very first drug to treat HIV infections. So if you take a look, AZT, it looks like thymidine. So it's almost like thymidine in every aspect, except for this little part over here. It has a different functional group at the bottom. So it turns out that AZT binds the HIV enzyme reverse transcriptase. So it binds a viral enzyme really tightly and does not bind human enzymes very tightly. And so this is this was, like I said, the very first anti-HIV drug that came out in the late, I think this was the late 1980s, and was revolutionary because, of course, before then there was no treatment for people with HIV. So lots of inhibitors follow this category and they're drugs. I'll show you a couple other inhibitors that are drugs. Here's another inhibitor. Maybe you know this one. I purposely left its common name off here to see if anyone would recognize it. This is ASA, which is acetyl salicylic acid. This may be the most commonly taken drug, maybe after caffeine, assuming you call caffeine a drug, taken on the planet. And you can see it says here it binds reversibly to the enzyme cyclooxygenase. So you're probably still wondering what it is. And there it is. This is aspirin. And so if you take aspirin or Tylenol or something like that, Tylenol is actually a slightly different compound. These are binding enzymes that are involved in the pain and inflammatory responses and are, like I said, the most commonly used drugs on the planet. We also talked about penicillin. So there's penicillin, and you can see it says here that this binds to enzymes involved in peptidoglycan synthesis. So we did mention this before way back in topic 5, that this affects enzymes involved in the cell wall, and then basically the cell wall is left with holes. So here's the question. It says, what would happen if a bacteria has penicillin and it's in a hypotonic environment? Well, you get holes in the cell wall, and then the cell ruptures and releases its contents. So this is how penicillin kills bacteria. So some inhibitors can be bad. Here's an example of one. This is called serine, and this binds to serine, which is important for an enzyme called acetylcholine esterase. So you may or may not know what acetylcholine is. Acetylcholine is a neurotransmitter. So this here is a neurotoxin and is obviously very bad for us. This was actually used a number of years ago. A terrorist group released some of this nerve gas in the subway in Tokyo, and a number of people died from it. So not all inhibitors are this bad, but they are blocking enzymes, and depending on what enzymes are being blocked, it can be therapeutic, it can be toxic, or they can also be used to regulate activity as well. And some inhibitors are actually found naturally in your body, by the way, and this is what I'm getting at and what I want to talk about in terms of glycolysis in a minute here. So before I talk about glycolysis, I want to talk about something called allosteric regulation, and I've sort of already hinted on this already. So allosteric regulation, it says here, is when there's some sort of regulatory molecule and it binds one part of an enzyme and it affects another part of the enzyme. So it's kind of like the non-competitive inhibitors, but it doesn't necessarily have to inhibit it. So if you take a look, here's an enzyme, and just in equilibrium, enzymes are relatively flexible. So I think thinking of an enzyme sort of like a palm of your hand is actually a good analogy, right? And if you're playing baseball or if you're catching a frisbee, there are certain conformations in your hand that are more likely to catch that frisbee than others. Like if your hand is closed, you're not going to catch that ball or frisbee. But if your hand is open a little bit, it's going to be in the perfect shape to catch the frisbee or ball. So what these allosteric regulators do is they can inhibit. So I mentioned that one already. Maybe my hand is closed and it's closed because I'm wearing a mitten. And so I'm not going to catch a frisbee very easily with a mitten. And that's what an inhibitor is like. Sometimes they are activators and they actually stabilize the active form. So maybe hold the palm in the right place in the right angle, and they can interact with the substrate. So this is what allosteric regulation means. It's binding one part of the molecule, and it's either making the molecule ready to bind something or maybe not as efficiently. And like I said, I have an example here that I want to give you in glycolysis, but I just have a couple more slides to cover first, so just bear with me, please. So a lot of pathways as well have feedback inhibition, and you've probably heard a little bit about feedback inhibition in high school. This is the case with a lot of hormones and things like that. Our body, when it realizes it has enough of something, that end product goes back and tells an earlier stage to stop doing it. You can see here's an example of a biochemical pathway. And in this biochemical pathway, we're going from threonine, that's an amino acid, and we're making isoleucine. So this has 5 enzymes, so that's 5 chemical reactions. And in order to do that, it's going to take a little bit of energy, and it's going to consume cellular resources. So it turns out what happens is our body has this pathway, and those enzymes are functional, and we make isoleucine. But if we end up with a lot of isoleucine, then what happens is that isoleucine goes back and it binds this enzyme over here. So notice there's that extra little binding pocket, and what it does is the isoleucine binds that other pocket and affects the active site, and so it turns off that pathway. And this is called feedback inhibition, where an end product, like I said, it goes back and turns off an earlier stage of a chemical pathway. If the isoleucine gets used up, it eventually diffuses away, and we end up with that pathway basically getting turned on all over again. So that's what feedback inhibition is. And like I said, I wanted to show you what's going on with glycolysis. Sorry, we'll come to Kahoot in a moment. I'm going to just draw you a picture of a little section of glycolysis. So I'll go find my whiteboard. Here's my whiteboard. Okay, so I call this allosteric regulation in glycolysis. So let's talk about what's going on in glycolysis. So we're going to talk about glycolysis in the next unit, but it's worth introducing some concepts now. So glycolysis, by the way, you probably know glycolysis is a biochemical pathway, and we start off with glucose. So that's where we're going to start off with glucose. And so it turns out that we have a couple of reactions. I'm not going to draw these reactions. I'm not going to draw the structure of glucose for you, but if you want, we can do that sometime, but I'm not going to do it now. I'm going to just draw in a couple of things, and I'll come back to them in a second, but there's basically 2 reactions, and then we get a compound called fructose 6-phosphate. Okay, and fructose 6-phosphate is catalyzed, and we get fructose. Oops, I'm going to get that eraser. And then we get fructose 1,6-diphosphate. And that's the reaction I want to talk about, but I'll come back to that in a second. So like I said, there's actually 10 reactions in glycolysis. So I'm just going to call this several more steps. And we're almost at the end. We get something called PEP. PEP actually is an abbreviation for phosphoenol pyruvate. And then one more step. And we're going to get to something called pyruvate. So you probably remember from high school that glycolysis is the reaction where we're taking glucose and we're breaking it down to pyruvate. So glucose, by the way, is 6 carbons, and we're actually going to get 2 pyruvate in the end, but I'm not really worried about balancing chemical reactions right now. What I want to talk about is what is going on here. And so specifically, what is going on right here is we have an enzyme called phosphofructokinase. I always laugh at this enzyme because phosphofructokinase, the abbreviation is PFK. And I ask people where they've seen PFK before, and usually at least one person gets it. PFK is the acronym for Kentucky Fried Chicken in French. So it's poulet. I'm not sure what the equivalent is in French. Anyway, phosphofructokinase is that enzyme, and that's the one I want to talk about. It turns out phosphofructokinase is one of these regulatory allosteric enzymes. So what is going on here? Well, I'm going to draw some arrows and a couple of other things. Let me see here. Maybe I'll try a green pencil this time. And so I've mentioned this PEP compound here. And so this is not quite the end product of this pathway. It's almost the end, but it's more useful for various reasons because glycolysis, these are not the only reactions involved in your cell. And so it's more useful for the cell to actually regulate using PEP. So PEP, I'm just going to draw an arrow like that. So PEP is an allosteric inhibitor. Allosteric inhibitor. So that means if there's enough PEP in the cell, it goes back and interacts with that enzyme, phosphofructokinase, and turns it off. There's one other thing, and I'm going to write a little note on the side in a moment here. There's one other thing that I wanted to mention, is that ADP, it turns out, is an allosteric activator. So I'm just going to write some notes on the side here. Let me just move this over here, small to make that. Hopefully you can still see it decently. And let's just make a few notes about this whole thing, okay, what all this means. So let's start off with ADP, okay? So think about this. So if we have ADP, and I'm putting the square brackets around it, that means ADP concentrations, right? So if ADP concentrations are high, what does that mean if we have lots of ADP? Well, if we have lots of ADP, it probably means we don't have lots of ATP. So that's the first thing to consider. So this means that ATP levels are low. So this is a problem because the cell wants ATP. So if we have lots of ADP, it means we want to turn this whole thing on. So hey, let's turn on glycolysis. So that's what the cell wants. So I'm going to write that down. It means the cell needs more glycolysis because glycolysis is going to make ATP, right? This is what it's all about the mighty ATP. And so therefore, phosphofructokinase is, and the term we use for turning things on means upregulated. That's the term we use for turning things on, upregulated. And of course I can't spell because I'm talking. This keeps happening to me. Let's try that again, upregulated. And like I said, that means turns turned on. Okay, so, and this should be a D, not a P. So I'm sorry about that. My D's and my P's have always looked too similar. So that should be ADP. So what about that other compound there, PEP? Let's just write a note about that. So if PEP, so phosphoenol pyruvate, so if those levels are high, what does that mean? It means we have a buildup of glycolysis products. It means we've had a lot of glycolysis and those products are lingering and sitting there. So let's call that a buildup of glycolysis products. Glycolysis products. And then I'm going to put in brackets including ATP. Or presumably. It's not a perfect system because you could have lots of PEP and maybe not lots of ATP, but you know, this is the system the cell has developed. It is more complicated than this, but this is kind of the basics for which are good enough for this class. So what does that mean? It means we want to stop glycolysis. So the cell wants to stop or slow down glycolysis. So what does it do? Therefore, it downregulates the enzyme. Downregulates the enzyme phosphofructokinase. Okay, so we are going to come back and we are going to talk about glycolysis, and we are going to, I'll mention phosphofructokinase again as well. So we have just enough time, I think, for a Kahoot. I have 8 questions. Let me load that up for you. You can load up your app, and then that will wrap up today. Okay, I thought this would load up for me. Something's going on my end. Just give me a moment, please. Maybe my internet connection is a little slow or something. Okay, well, I'm not sure why this is not loading up. Maybe it's on Kahoot's end, so I'm sorry about that. What I will try to do at some point before the next midterm is I will send you a link that will have all the Kahoots so you can review them. So sorry I couldn't get this one working. I'll make sure I have one for you guys next day. But that's all I want to talk to you about today. Wednesday we're going to talk about respiration, and that's also going to spill over to Friday. And then maybe Friday we'll talk a little bit about photosynthesis. We'll see where we get. And then we have the cell cycle. And I know I'm running on. It's a long run-on sentence. I'm just warning you that we are slowly approaching midterm 2. It's going to come a lot quicker than you think. So just a little heads up on that. Anyway, that's it for today. I will see you on Wednesday.